I'll just share my screen. So I'm going to talk to you this morning about one of my favorite topics actually, which is, okay, hopefully you can see the presenter view there. Neuroprotection in the injured CNS. And I'm particularly going to talk to you about the eye here, because obviously I work on spinal cord and the brain as well, but some of the things that we learn in the eye can be directly translatable to the spinal cord and the brain. So what I've been working on in Birmingham with Professor Anne Logan, who's also on this call somewhere, and Richard, of course, and many other colleagues here, is what are the main constraints on regenerating a damaged axon, and specifically central nervous system axon. And one of the top things that comes up, and there are about 4 or 5 things, but one of the top impediments is neuronal cell survival. And this is why I've highlighted on this slide in red. There are also lots of other things like the deposition of a glial scar, which contains lots of inhibitory molecules like semaphorins, ephrins, chondroitin sulfate proteoglycans. So you will have to deal with the glial scar at some point. Also, the degenerating myelin releases lots of epitopes that are inhibitory to central nervous system axons. So you're going to have to deal with no-goes and oligodendrocyte myelin glycoprotein, myelin-associated glycoprotein. But what we've learned is that if you don't have enough cells that are surviving, you might as well not bother with the rest of them, because if the cells are dead, you can't replace them. And so what we're really interested in our lab is how do we promote survival? And I'm going to talk to you about some of the ways. There are lots of different ways. There's another person on the call, Roshia. She's done a lot of work on antioxidants and stuff like that. And I'm actually doing a systematic review with one of my students on neuroprotective drugs in the last 10 years. And Roshia's work features quite heavily in that. So Richard's already told you this, but this is my particular favorite model, the optic nerve injury model, developed many, many years ago and first shown by Professor Berry, an emeritus professor with us. In 96 and 99, published some beautiful work. So you have to the left of your picture, you have the retinal ganglion cells. They project their axons, which forms the optic nerve, and this reaches the chiasm. This is a great population of neurons to study because all of the axons go in a single direction. Whereas if you remember, the spinal cord is very different. Things go up, things come down, things go across the midline, et cetera, et cetera. So when you want to target a specific population of neurons, it's very difficult to. Whereas here, you can pretty much put about, in a rat, about 10 microliter of fluid into the back of the eye to do an intravitreal injection, probably around 5 microliters in a rat, in a mouse, sorry, and probably up to about 30 microliter in a human. So what we do is we make a small lesion behind the eye, which leaves you a proximal segment and a distal segment. And you can sort of put traces in and work out how many cells are surviving and how many are dying, how many have survived and regenerated through the lesion site, et cetera, et cetera. And here's just a bunch of publications that I've put on there, but there's lots more. The biggest problem we have is that retinal ganglion cells, these are just one population of the retina that we need to protect from dying, die very, very rapidly. So within 7 days, 40% of the cells are dead. Within 14 days, about 80% of the cells are dead. And by 28 days, the majority of the cells, if not all of them, have died. So this is what Richard was talking about, and I quizzed Richard about it on this in terms of timing. Obviously, after an eye injury, a traumatic optic neuropathy, you're going to have to get your treatment in fairly quickly. Otherwise, it's no point doing it 10 days later or 2 weeks later. You're going to have very few cells in order to promote regeneration, if you were to promote regeneration. So really, the ultimate aim is neuroprotection. But the point of neuroprotection is I want to see a picture like this, for example, the bottom picture. So this is a non-regenerating optic nerve. This is a section through the optic nerve, through the lesion site. To the left of our picture is the eye. To the right of our picture is the optic chiasm. And these axons are growing from left to right. In green are growth-associated protein 43 to stand for regenerating axons. And you can see that in the non-regenerating model, very few axons are growing through the lesion site, and in fact, growing up to the lesion site. Because here we only have about 10% survival. These are assayed at 21 days after injury. However, if you look at our regenerating model, and I'll come back to the regenerating model, we have 60% RGC survival here, approximately. But you can appreciate lots and lots of GAP43 staining in the proximal segment, lots growing through the lesion site and finding their way into the distal segment. So these are actually regenerating. So the point of this slide is to show you that you need some RGCs available there for you to promote this sort of regeneration. So a very first problem is can we protect these neurons from dying, and if we can do that, we might be able to preserve visual function. Why do neurons die? Well, neurons, as you know, retrogradely transport their neurotrophic factors, which is what keeps them alive and which is what helps them to regenerate, back to the cell body. So they retrogradely transport stuff to the cell body. If you interrupt that, there is no way that they can get the neurotrophic factors back into the cell body. And because they're unidirectional, these axons, you cut it, there's nothing. Whereas in the spinal cord, if you cut the spinal cord pretty much anywhere, they may still be alive, and there's very little cell death in the spinal cord because of the fact that they receive inputs from multiple areas. So they can still receive growth factors from elsewhere. Whereas the optic nerve or in the eye that we're dealing with, as soon as you've interrupted that supply of neurotrophic factors, that's pretty much fairly catastrophic. You need to try and get that back up quickly. And obviously your local glia, they do express and secrete some growth factors, but they'll only do that for a very short amount of time, for about 7 or 8 days, and then they stop producing growth factors as well. And obviously these neurotrophic factors are really important because they maintain the integrity of the cell, but also they promote their regeneration as well. So pretty key compounds. One of the ways, or a couple of the ways the cells die are necrosis and apoptosis. And I've talked to you about apoptosis a lot more in my previous presentation to the Orbital seminar series, but I'll just mention a few points. So necrosis is just plain, depending on how much insult you have, it's just a massive death of everything, and there is no regulated order essentially. Whereas apoptosis is much more ordered, rounding up of cells, retraction of pseudopods, nuclear fragmentation, etc., and then you get blebbing of the plasma membrane, and then engulfment of resident phagocytes. The point about apoptosis and necrosis is that necrosis, because everything explodes in the surrounding area, it causes a huge amount of inflammation. Apoptosis, because of the way it's regulated, does not cause inflammation in that particular area. So that's a key difference between necrosis and apoptosis. But when you look in the literature, there are lots of different types of ways the cells die. So we've actually dabbled a little bit on necroptosis with Richard. There's autophagy, mitophagy, ferritinophagy, and pyroptosis. These are all becoming very, very popular, and these are sort of many other ways that cells die. But if you read most of the literature, the majority of the ways are necrosis and apoptosis. These are sort of additional ones that you can target. So I'm not saying that apoptosis is key, but there are lots of other pathways to target, and opportunities exist here as well. Caspases are one of my particular favorites. So there are 12 different caspases, mammalian caspases, and they all have this sort of structure. And if you want to read more about it, there's, I think, a 50-page review that Vasanthi and I wrote last year, published in Cell. So you can go back and read that if you want to. But essentially, we're looking at caspases that are grouped into 3 different groups. They're sort of caspases with long motifs, slightly shorter motifs, and ones that are execution of caspases, group 3 here. Group 1 are mainly initiator caspases. Group 2 have recently been shown, and I'll talk a little bit about caspase 2, to have a bit of both, a bit of executioner and an initiator motif, whereas group 3 caspases only have an executioner motif. So if you read the literature in the past, most of the literature would say that the neurons die by caspase 3. And then later on, it was shown they die by caspase 6 or caspase 7 mediated mechanisms. In our laboratory, we've shown that caspases pretty much, retinal ganglion cells especially, die by caspase 2 mediated mechanisms. The activation of these caspases is also very, very complex. Looking at caspase 2 is the most evolutionarily conserved caspase amongst all of the populations of caspases. But they have very, very complex activation motifs, and they are activated in a variety of different pathways. And this is why they're very, very difficult to study. For a very long period of time before we showed our work on caspase 2, it was not common that caspase 2 could act as an executioner of caspase. But we were able to show, we were one of the first to show that caspase 2 can act as an executioner of caspase. And if you block caspase 2, we can pretty much block a lot of the apoptosis that goes on in the retina. When it comes to apoptosis, there are two main pathways. There's the extrinsic pathway and the intrinsic pathway. The intrinsic pathway, something catastrophic happens, the mitochondria depolarize, they release their cytochrome C, and that activates caspase 9, which then converges on caspase 3 eventually. And caspase 3 is the execution caspase, caspase 9 is the initiating caspase. You've also got the extrinsic pathway. So something catastrophic happens, release of factors, these bind their particular receptors. They involve caspase 8, but eventually that also cascades down to caspase 3. And caspase 3 is the execution caspase that causes DNA fragmentation, membrane blubbing, and eventual cell death, because this is very integral to the maintenance of that cell. Some of the targets of effector caspases are nuclear, DNA-related, cytoskeleton-related, cytoplasmic, and also protein kinase related too. So there are a variety of factors that are involved in this process. And you can see that all of the different targets. I won't bore you with all of that, but just to know that there's lots of different targets for these sorts of things. What we've shown recently is that in the CSF itself, after an optic nerve crush injury, you can detect these caspases too. And this is quite interesting. So if you look at the pathway, so within 5 hours, we see activation of caspase 2. The other ones are quiet at the moment, but after 24 hours, you can see we've got more activation of caspase 2. But now at caspase 6, 3, and 9 are increasing. At 7 days, you've got increase in caspase 2, 3, 6, and 9. By 28 day, caspase 2 expression completely is gone, but caspase 3, 6, and 9 remain. If you look at the activity of these then, so that was just actual levels. If you look at the activity, that mirrors the levels of caspases. So you can see the caspase 2 activity is quite high within 5 hours, peaks at 24 and then starts to diminish. The other caspases, 3, 6, 6, and 9, start to come up slightly later on, and they remain fairly high up until 28 days after injury. And this is in the CSF, so this is way away from the original site of injury. What we also see along with this is that caspase cleaved products are also found in the CSF after an optic nerve crush injury. And you can see all of these different proteins, caspase cleaved cytokeratin, caspase cleaved tau, alpha spectrin, SDP120. These are sort of typical ones that caspases cleave as soon as they're activated. Within a few hours, you've got CCK18 activated, that peaks at 24, and then you see this temporal expression. And what this is showing you is that there are lots of different caspases that are activated at different time points after injury. So although we've targeted caspase 2, you may actually need to target other caspases too in order to protect other populations of neurons. In terms of caspase treatment then in the eye, we surveyed the literature and published a paper in 2017. You can see that if you use inhibitors of caspase 1, 3 and 6, 6, 7, 8, and 9, you get pretty good neuroprotection. So the best one was caspase 7, which gave you 76% neuroprotection. But if you look at caspase 3, which most of the majority of the literature suggests is the one that kills neurons, only 35% of RGCs are protected by using anti-caspase 3 treatment. So we in our lab reason that there must be other caspases that are involved that protect a lot more. Hence our view of looking at caspase 2. We know that we published a paper in 2006 showing that neurotrophic factors, if you give them in combination, not individually in combination, they can synergistically promote survival. And you can see that when we gave FGF, NT3, and BDNF, so fibroblast growth factor, neurotrophin 3, brain-derived neurotrophin factor, we could get up to about 43%, nearly 45% RGC survival. So it looks like a combination rather than them on their own or in doublets gave you much better survival. So that's one strategy. Obviously, finding a role for caspase 2 was pretty difficult. It was shown in the literature in around 2006, 2007 that ER stress activated caspase 2, and that caspase 2 could then cleave nuclear proteins. And this was surprising because it was always thought that caspase 2 is an initiator caspase. So we started looking at it and got very interested. And so we looked at our sections of eyes after an optic nerve crush injury. And one of my students came back to show us this. On the left, we have staining in green for cleaved caspase 2. So this is activated caspase 2. This is the ganglion cell layer, and these are the photoreceptor layer at the top of the picture. But you can see that the majority of the ganglion cells were suddenly lighting up with staining for cleaved caspase 2. So we were pretty surprised by that. When we looked at cleaved caspase 3, unfortunately, this is in red now. So you're looking for red staining in this layer. In the ganglion layer, there were very few cleaved caspase 3 positive cells. So we weren't convinced that caspase 3 had that much to do with ganglion cell death. It does play a part in the lower layers. The inner nuclear layer is stuffed full of caspase 3 positive cells. So it might be relevant to those particular sets of neurons. I'll skip that slide. But at that time, we delivered an siRNA to caspase 2 with the help of a company which developed this product, which was very long-lived and it was stable and resisted nuclease degradation. And it was an siRNA sequence against caspase 2. And if we delivered approximately 20 to 35 micrograms of this compound into the eye after an optic nerve crush, we could pretty much protect all of the ganglion cells from dying. So this was phenomenal, actually, that we could protect pretty much all of the ganglion cells using this siRNA to caspase 2. So we were convinced that caspase 2 definitely has a part to play in this. And obviously we showed in another publication later that optic neuritis, which is another form of eye disease where you get about 30% 30% death of retinal ganglion cells. If you use caspase 2, we could protect the majority of those. Whereas if you use a control siRNA, you can see quite significant cell death. So it looks like we can use it in not only in traumatic optic neuropathy, but other areas where you get death of ganglion cells. But as we discovered, just because we could promote pretty much 95 to 98% RGC survival, didn't mean that we would get axon regeneration. So we know that survival doesn't equal regeneration. So although we can protect them from surviving, we need to add additional compounds in order to promote regeneration of these axons. And here what we did was we combined the caspase 2 with the caspase 6. It's been shown by a different group that caspase 6 can promote some axon regeneration. And lo and behold, we did have some axon regeneration in the optic nerve after a crush injury. So it looks like caspase 2 is doing its job. We took at the moment we deliver caspase 2 every 8 days in our animal models. But we took this out to 12 weeks, delivering it every 8 days, and we could see that if we give our caspase 2 treatment, we can protect upwards of 95% of our ganglion cells. So it looks like as long as we give our treatment fairly quickly, so we give it at day 0 and then every 8 days, we can protect it for a very long time. And we haven't taken it out beyond 12 weeks, but I suspect we could probably go a lot more beyond that. And something that I work with Richard on is blunt ocular injury, so other forms of ocular injury. And so here, what we did was develop a model. Richard developed this model as part of his PhD, and then one of our PhD students looked at it. And we find that caspase 2 is once again activated in these different models of ocular trauma where you get death of ganglion cells. So it looks like ganglion cell death could be related to the expression or the upregulation of caspase 2, which is really interesting. This caspase 2 is now in phase 3 clinical trial. They've already done a couple of clinical trials and they haven't produced very useful. They haven't met their primary outcomes because they decided they were going to start treatment in 2 weeks after patients presented with their disease symptoms. And as Richard has mentioned something about timing, and this is why I quizzed him about timing, it looks like from our experiments that the caspase 2 treatment has to be given very quickly. The quicker the better, because within a couple of weeks, 80% of our ganglion cells die in our rat model. So we have to give them very quickly. So it might be that they've chosen the wrong model to study this. They are doing glaucoma trials in glaucoma at the moment, and they're recruiting patients. So we'll wait to see what happens there. And the other interesting thing that we found a few years ago is that when you give other neuroprotective agents like pigment epithelium-derived factor, which can promote about 80% survival of ganglion cells, we showed that this was by suppressing caspase 2 eventually. So it might be that there's a common pathway where things that are neuroprotective might actually work through caspase 2. BDNF is another really good neuroprotective agent, but that was also shown to work through suppressing caspase 2. So it might be that all of these go down to caspase 2 eventually, because caspase 2 is the execution of caspase for ganglion cells. Something that I've worked on very recently are DNA damage inhibitors. So these are inhibitors to sense damage in neurons. And as soon as we found that as soon as you cause an optic nerve trauma, you increase this damage that the neuron accrues within itself, within its nuclear membranes. And if you use specific drugs that enable the cell to recognize these bits of damage to their DNA, you can protect these ganglion cells from dying. So in an in vitro model, if you give these drugs which inhibit DNA damage, different pathways, you can get pretty much 80% survival, 90%. In vivo, we use these molecules and we get about 85% neuroprotection. But recently I've shown that even though the DNA damage inhibitors protect ganglion cells, it actually does it through suppressing caspase 2 eventually. So although they work on themselves, they are having an impact on caspase 2. So although I've come to the summary now, although I've labored on caspase 2, don't forget there are lots of other different treatments. You know, there are antioxidants, there's GSK3, lots of different pathways that do mediate cell death. But I propose to you that eventually most of the pathways that I've worked on come down to caspase 2. They impact on caspase 2 in some way, and that might be the reason why we get a neuroprotective effect for retinal ganglion cells specifically. So just to summarize that, in order to promote regeneration in the first place, we're going to have lots and lots of cell survival. So cell survival is absolutely fundamental. And obviously your ganglion cells, you can't replace them at the moment. There aren't any treatments that replace everything. So we need to try and keep alive as many as possible. And I've talked to you about the timing of intervention is really, really important. And we think that caspase 2 is a key caspase in the death of ganglion cells. Richard has shown, and I haven't talked about this, but for example, caspase 9 is required for photoreceptor death. So it looks like different populations of the retina require different caspases and respond. Certainly the ganglion cells we think respond particularly to caspase 2. And we think that DNA damage inhibition might be a novel neuroprotective strategy. But obviously the problem is, and this is what you guys are working on, is how do we deliver some of these potential treatments safely and effectively, and obviously timely as well. Thank you for listening. I'll take any questions.
Thanks, Zubair. Fantastic talk. As we don't see any questions in the chat, and there's nobody speaking up with their hand up yet, I'm going to monopolize you for a little bit. Please do anybody else put your hand up or put it in the chat if you're going to ask questions. So first question, Jenny, you've covered this a bit already. If we look at attempts to translate neuroprotective therapies over the years, we've got the recent studies that you referenced, so the NARN study that's been stopped early because intermittent interim analysis shows it wasn't probably going to meet its primary outcome measure. We've got the, and I'll exclude the studies where they're treating the cause. There's lots of good examples of neuroprotection, like in glaucoma, lowering IOP, like in traumatic brain injury, stopping hypoxia, so stopping secondary injury, treating the cause, but actually interfering with cell death pathways. And there's memantine studies in glaucoma, in Alzheimer's, in stroke, none of which have shown any benefit to neuroprotection. Why do you think there's been such an abject failure over the last 20, 30 years to translate any neuroprotection?
So that's a really good question. It's obviously a very long-winded answer for that. And I think one of the main reasons for most of these trials failing is timing, because we know that if you're trying to protect anything, the cell death pathway machinery is activated very, very quickly. Within 5 hours, for example, caspase 2 is very high. Some of the other caspases are very, very high as well within that particular time. By about 10 days, the majority of the caspases have activated, done their business. For example, if you look at a rat retina, after about 7 days, you don't find any tunnel positive cells anymore, because the process is already done, it's activated, and then it's all the secondary degeneration. Most of these clinical trials, they've tested the drug very late on. So for example, the NAOI, the treatments that you talked about with the caspase 2, they started treating from 2 weeks onwards. And to my mind, that's way too late. So you need to treat very, very early. If you can, within 24 hours would be the ultimate thing. And that way you can save many of those things.
Yeah. Okay. So I guess the other possibility, and this is probably a bit of a devil's advocate question. Do you think we're protecting something functional? So you've got a cell that's had cell death pathways activated. Are you protecting a cell that can work and is functionally connected to its targets?
Yeah, I mean, from our studies, so we've done a little bit of electrophysiology. They certainly respond in ERG studies. So, and I guess the thing to do is, if you want them to be ultimately functional, is to regenerate them back to their targets. And then you can look at visually guided behavior and whether that is actually functionally useful then. But obviously for that, you need a good regenerative treatment to take it all the way back to the chiasm and the visual centers. So I think we've still got a very long way to go, and there's loads of opportunities for you guys that are in this room to come up with some really nice neuroprotectants. But one thing that's come very, very clear from our studies is that you have to get your treatments in very, very quickly for neuroprotection. The cells activate cell death pathways very rapidly. Whether you look at any of those, whether it's pyroptosis, necroptosis, or any other optosis that will come about in the future, you're going to have to get your treatments in very, very quickly.
Yeah, so we've got a question now from Aya Byrne. Thanks, Zubair, for the great talk. What's your opinion on caspase 7? Can caspase 7 give better clinical efficacy? Also wanted to ask how you delivered siRNA against caspase 2. So I guess caspase 7 and initiated caspase, should we be going upstream or downstream?
Well, caspase 7 is another effector caspase. So it's an execution of caspase, and it's pretty good as well. It gives you about 60 to 70% neuroprotection. The problem with a lot of these caspases is that they lose their potency the longer you leave them out. So, for example, the initial study with caspase 7, they were done up to about 14 days. Abbott Clark did them in the US. When I communicated with him, and by about 12 weeks, that it loses its potency. So then you don't protect anymore. Whereas when we took out caspase 2, for example, out to 12 weeks, we still have 95% neuroprotection. So there is a clear difference. The other thing about caspase 7 is, like caspase 2, it suffers the same problem. It doesn't promote any regeneration. So yes, you'll keep your neurons alive, but if they don't have any axons to transmit the information, they'll be functionally inept, essentially, or useless. So you do need to promote regeneration as well as give them. In terms of delivery of our siRNA, we delivered it by intravitreal injections every 8 days.
Okay, and so final question, and if you answer this in the next minute, does blocking caspase 2 mediated apoptosis make cells cancerous?
Very, very good question. So when I've looked at neurons, I'm not interested in apoptosis, cancers. So it may do, but in the eye, caspase 2 has no other function at all.
Yeah, I would also caveat that answer before we move on by saying that we're usually aiming to give treatments for a short period of time, or forever. Yeah, if you knock down apoptosis over a number of years, that does really become a real risk. So our next speaker is Ben Mead, who previously worked